Synaptic communication is a process of communication between two nerve cells, aka neurons. The cell body contains a nucleus and dendrites. The cell body continues into the axon, which has a sheath of protection called myelin. The axon splits into axon terminals at the end, which create the synapse between another neuron's dendrite. When a neuron is connected to another neuron by the dendrite, the passing of the signals from the first neuron to the second neuron is called synaptic communication. An action potential starts at the base of the cell body. For a resting neuron, the intercellular fluid is higher in potassium ions and has a resting potential of -70 millivolts. When a stimulus occurs, it signals action potential to start depolarization. This is when the stimulus generates the action potential enough to open the voltage-gated sodium channels. This is called threshold. Sodium ions go from the extracellular fluid into the intercellular fluid until it reaches +30 millivolts. Then the sodium channels close. Repolarization occurs in the cell and opens the potassium channels to move potassium ions from the inside of the cell to the outside of the cell. The action potential moves down the axon due to propagation. When the action potential finally reaches the axon terminal, this neuron is now called the presynaptic neuron. The presynaptic neuron will take the action potential and make neurotransmitters that pass through the space, otherwise known as the synaptic cleft. This then goes through to the receiving neuron or postsynaptic neuron. This impulse opens the voltage-gated calcium ion channels. The neuron then engulfs the neurotransmitters that have been made. The neurotransmitters will then be dumped into the synaptic cleft through exocytosis. Synaptic delay is the time it takes for the nerve impulse to travel from the presynaptic neuron to the postsynaptic neuron. When the neurotransmitters reach the postsynaptic neuron, they bond with the ligand-gated ion channels and open the gates. The neurotransmitters directly bond to the ionotropic receptors. An excitatory neurotransmitter will cause depolarization, bringing it nearer to the firing of action potential. These open membrane channels for sodium and calcium ions. Inhibitory neurotransmitters cause hyperpolarization, making it more difficult for action potential to start. These open ion channels for chloride and potassium ions.